Hello, everybody. Welcome to the Kids First fall webinar. The topic will be empowering pediatric research with data innovation. And I will welcome everybody and thank you for being here with us today. I'm Marcia Fournier, Kids First Program Manager. My education in cancer biology and clinical oncology brought me to the program a little bit less than 2 years now. And I will be your master of ceremony today. And we'll talk a little bit about the program and funding opportunities. The agenda today will cover excited topics. We'll share with you an exclusive show of a new video about Kids First, produced by the Common Fund. And following, I will talk a lot about the program impact and funding opportunities. And then we will hear from JP on Kids First portal feature. Dr. David Higgins will demo the Kids First portal, and followed by Sean Griffith from Johns Hopkins University, who will demo a new tool linked to the platform CAVATICA, PhenoDB. And to finalize the program, we will hear from Dr. Adam Resnick on his vision for the Kids First Data Resource Center. And we will have questions and answers in the end, but also feel free to put your questions in the chat box. We'll be sharing links in the chat box, so check it from time to time as we're going to be sharing relevant links for you. As I said, we will start today showing this video produced by the Common Fund. Is a new video about the program, and you are seeing it. It's shown for the first time today, so you have the exclusive preview of this video, which will be later shared with the broad public. But whoever is here today, thank you for joining. I see that people are still joining. Will have the privilege of seeing it first. So I will start with the video and I will come back. And as I said, today the topic is showing the power of innovation through data. And my goal here today is to help you to discover resources that the program is making available to researchers globally. So let's start with this show. I'm eager to show you. I will stop sharing for a moment as I can get the video here. I will share again. Oh, actually, here we go. And I hope you enjoy the video. We'll be back in 3 minutes with you.
Did you know that the leading causes of death among infants and kids are birth defects and cancer? The NIH Common Fund's Gabriella Miller Kids First Pediatric Research Program, or Kids First, aims to help researchers uncover new insights into the biology of childhood cancer and structural birth defects, with the help of children and their families. This program was inspired by childhood cancer advocate, 10-year-old Gabriella Miller, who was diagnosed with inoperable brain cancer, and who boldly challenged the United States Congress and research community to stop talking and start doing. Children with structural birth defects, including congenital heart defects, orofacial clefts, and neural tube defects, also have an increased risk of developing childhood cancer. This suggests that there may be shared genetic pathways underlying some childhood cancers and structural birth defects. The Kids First program is creating a data repository where data from children and their families with these rare conditions are available in one place, where they can be accessed, analyzed, and visualized. Researchers collaborating with children and their families can apply to submit genetic samples for research use by Kids First. These samples undergo whole genome sequencing. Genome sequences from participants are harmonized and put into a large-scale data resource where researchers can access a variety of information that has been made ready for public use by investigators and the Kids First program, including genetic data, which includes the genome sequencing, as well as clinical data from patients and their families. Analysis and visualization tools are also available through the Kids First Data Resource Portal. These tools help researchers understand the data in new ways. By understanding one condition or comparing data from several conditions to look for common pathways. This allows researchers to make new connections between genetic pathways found in both structural birth defects and childhood cancer. Discovery of these new connections have the potential to lead to a better understanding of disease mechanisms, leading to more effective disease prevention, diagnoses, and treatments. As of early 2023, Kids First has investigated 1,940 unique health conditions, while supporting 63 sequencing projects, which have made nearly 25,500 participant genomes available to researchers around the world through its Data Resource Center portal. This has led to new scientific discoveries for Down syndrome, cartilage tumors, vascular anomalies, and more. These collaborations have the potential to lead to new prevention methods and treatments, alleviating suffering for many children and their families. Are you interested in learning more about the Kids First program? Visit commonfund.nih.gov/kidsfirst. To access the Data Resource Center portal, visit kidsfirstdrc.org.
Okay, so I hope you all enjoyed that. And as I can't see you, I would love to see some comments on the chat, how you like the video. This is a newly produced video by the Common Fund, and soon will be available to the public, but today you had the exclusive preview of this video. Thank you so much for your warm claps. This way I know you all are there, and I will give you a short presentation, but the video said everything, so I will just complement this video with a little bit more details on the resources the program is making available to the research community. I want to then show you what we're doing with the Kids First program appropriation. As you know, the Kids First was a authorized program, a bipartisan program authorized in 2014 with its first appropriation in 2015. And since then, we've been building resources to make data available to the pediatric research, specifically in structural birth defects and childhood cancer. And from 22 to 24, which will be the last year of appropriation of the program, we are focused on generating data, continue generating data, not only genomics, but also proteomics and other types of data, and developing the data resources. And today you're going to hear how we'll have a demo with multiple tools now available in the data resource portal. And we also have been focusing on supporting expert-driven activities through data analysis, workflows, and tool development. How are we doing that? Kids First is empowered by the EXO-1 mechanism, genome sequencing centers, and data resource centers. But really, always starts with patient cohorts and your proposals. The Kids First resources, I want to tell you how it works. So we start with your cohorts, your pediatric cohorts and your proposals. And the Kids First program is being using a mechanism called EXO-1. The EXO-1 mechanism provides support to researchers through an in-kind support, giving researchers access to sequencing centers where you can get genomic data from that cohort you're submitting. So there are a few things I want to emphasize about the EXO-1 mechanism. If you're a researcher and you're doing pediatric research in the US or globally, you can apply for this mechanism. And the NIH expects that EXO1 will be published, the Kids First EXO1 will be published in this fall, October, November timeframe, so keep eyes on it. Few things to keep in mind, we usually prioritize that brings allow broad data sharing, and also brings rich phenotypes and clinical data associated with the cohorts. And so that is a big hint. So if you're writing an EXO1 proposal, emphasize how broad can the data be shared, and also which phenotypes and clinical data associated with that cohort that you can make it available. So once we prioritize, and this year we have 7 new cohorts prioritized in 2023, investigators send DNA and RNA for the sequencing centers, Broad Institute and Hudson Alpha. And the sequencing centers will generate the data, and once the data is ready, the Data Resource Center, and you're going to hear a lot about the Data Resource Center today, will compile the genomes, phenotypes, and clinical data and perform a quality control, which is a highly collaborative process between the investigators, NIH, sequencing centers, and Data Resource Center, to get this data ready and approve it by the NIH to be delivered back to investigators. So here you get your data, and now you can run with your data, create new hypotheses, and I will tell you a little bit about funding mechanisms that can support the data analysis. But also, after 6 months, this data will be made available in the Kids First portal, which now the data is available not only to investigators, but to all researchers globally. More often than not, after investigators get their data, they also, this can be hypothesis creation for new EXO1s. So many of our investigators have multiple EXO1s, in addition to new researchers applying for it. So today, in the portal, you can, this map, the body map shows you different conditions where data is already available. And again, I want to emphasize that this is not only available to investigators that receive support from the NIH, but is available globally to the public through the Kids First portal. So I invite you to check the portal website and check the different data sets. So these data sets comprise over 27,000 genomes among children and families. And from 33 studies that have been released so far, we already have over 600 secondary users that requested data, and the data was approved. Because this data is made available publicly, but protecting the privacy of the participants through a controlled access. But it's an easy process. And again, I invite you to come to the portal and find out how you can get access to this data today. Give you examples of few data that was released this year in 2023. We're very proud of one of these, one of the projects that is a collaborative project, bringing together multiple pediatric conditions. If you are a researcher in pediatric, if you're researching pediatric research, you know that a lot of the childhood cancers and structural birth defects, they may also be rare diseases. So it's sometimes hard to have a cohort with significant number of children for a condition, but through the Kids First resources, researchers now can put that small cohort in the context of thousands of samples. And so this collaborative effort, again, are investigating 3 conditions: Down syndrome, heart defects, and ALL, pediatric ALL, and it has over 2,000 participants in this study. So I invite you guys to check it out. And the next slide. These slides will be made available through the Kids First portal. And I also want to provide the links of many other studies released this year, childhood cancer, and many 8 different studies in structural birth defects. So again, feel free to check. And also we invite this cross-condition research using this resource because we've reached a sizable data sets that are available now for your research. Next slide. So the Kids First portal usage, I want to show you a little bit of the results. So we are in 60 countries. Portal users are in 60 different countries. So emphasizing the global aspect of this program with over 195,000 data files available, over 4,000 portal users, and over 6,500 total portal logins. Next slide. Another way of showing results and whether this data is being useful for researchers is tracking publications that use Kids First data. And we've been tracking publications since 2018. And as you can see, the number of publications are growing with a spike now in 2023. So I think that tells us that this data resource is valuable for researchers globally. So keep doing this amazing work. And we're looking forward to see this even increasing the number of publications even further by making available not only data, but resources that you're gonna be able to see today that with tools and workflows available in the portal. The next slide. So the Kids First portal is supported by a cloud computing platform, CAVATICA, where you can compute, analyze, and share data. The next slide. One of the tools that I want to make you aware is a variant browser, genetic variant browser, where you can get summarized level of the variant data, query variants across studies, narrow down variants of interest, identify a number of participants with a given variant of your interest, and filter variants based on a gene name. And I want to highlight that this is all curated, so they are all annotated with genome location, which gene is affected, and how affects the protein. So it is a rich resource that I'd love to, again, invite you to explore. The next slide. And also now back to funding opportunities. So because this is all in the cloud, and we believe the cloud is the future for data and collaboration in the data science. And also now I think, as you're gonna see today through the demos, is accessible even for researchers that are not bioinformaticians or data scientists. I think that this resource now is available to people like me that are molecular biologists and cell biologists. So I'm really encouraged to explore this data in the cloud. And one of the things NIH is doing is for making it easier for you. If you haven't been done cloud computing yet, NIH is inviting you to apply for cloud credits, which is sponsored by NIH and give you the opportunity without costs for the cloud credits, for the cloud computing, as this is sponsored by the NIH, to have that experience in the cloud. And we already have approved 13 projects. It's a simple email, answer a few questions, and so go to the website and send us an email answering a few questions. Essentially, you have to have Kids First data and use CAVATICA as a platform. That is the two requirements. And then start using your credits. And the cloud credits also offer a follow-up after you finish your first program. So we really want to make it easier for you to adopt cloud computing and leverage the data available at the portal. Next slide. The Kids First is part also of a bigger data ecosystem. And it is interoperating with multiple data resources at NIH and outside NIH, like the Common Fund data ecosystem, the NIH cloud-based platform interoperability, NCPI, and others. The next slide. And so if you want to learn more, I want to invite you for the next event that we will gather. We will be there in person. So this will be an opportunity to meet the Kids First team and the Data Resource Center team and present a poster. If you have done work with the Kids First, we are calling for posters, which is open until October 5th. You don't need to be registered to the American Society of Human Genome annual meeting. You don't need the registration to participate, but you need to register to this event, and the links will be available in the chat if you're interested in and I will get some of the questions soon. So I want to finalize also talking a little bit about another resource, a funding resource. As I mentioned in the beginning, the EXO-1 mechanism, which is where we start the cycle of data, of generating data, it's meant for data generation. But then once you get the data, you need to analyze the data. So the Kids First also would like to highlight a couple of opportunities for data analysis. And if you have either the primary data, but also using secondary, if you're a secondary user and using data from the portal, you're welcome to apply to these opportunities that are relevant for the Kids First data. And the links, again, will be posted in the chat, but also this slide will be available at the Kids First website for the RO3 opportunities for that analysis. And I want to finish by introducing you who is behind the curtains. At NIH, I work with a working group across NIH, representing multiple institutions, including the National NICHD, MCI, and others. But we are also people, and I want to highlight some of the folks here in some of the Kids First in-person events. So if you want to meet some of these people, come to meet us at ASAG annual meeting. So with that, I will open for questions, and thank you again. And I will check on the chat here some of the questions.
I think there's
and there is
a great question asking about, what percentage of the publications are from the primary owners of the data that submitted them the sample versus secondary. And I think that's a really great question. And we're seeing more and more a trend towards sort of collaborative projects and expansion of the scope of work by both the primary investigators and secondary users. And it's something that we're going to go start tracking even better in the next iteration of our assessments. That's a good question.
Correct, Adam. And most of the publication so far has been the primary investigators, but also we are seeing multiple secondary user publications. So, okay, so submitted request for data access, it seems, and is being rejected. So we can take that to the committee that approves the data requests, and I can bring that up to our committee. But I won't be able to answer the question at this point. But thank you for pointing out, and we will bring that to the committee, that approval committee. Any other questions that Adam, that you see that you want to point out for us to answer now?
I see a question regarding patient communities who have their own registry based data and how to support it. I think this is an ongoing question, and I think increasingly we see sort of disease specific efforts looking for ways to intersect and connect with the Kids First infrastructure. I think there are several different mechanisms potentially for doing that, and we can review that maybe offline more directly. It looks like Dawn, feel free to reach out directly to the DRC, and we can discuss more specifics around that.
Correct. So again, a question that collaborate with researchers who submitted the patients for sequence, or can we work independently? So you can do both. We encourage collaborations, but we also encourage independent submissions. And if you talking about the EXO-1 mechanism, we will have, as we expect, again, that a new EXO-1 NOFO, which is Notes of Funding, will be published this year. So we're expecting the fall a new publication, and once that is published, we will have a pre-application webinar, and if you want more details on how to make your proposal competitive for EXO-1, I invite you to attend the pre-application webinar, which should be in this fall once the Notes of Funding is published.
Marcia, I think there's also a question regarding timeline expiration for cloud credits, with a question of whether or not, if someone has not used them within a period of 12 months, do they expire? And I don't think we may not have had that direct use case, but I suspect that, we would encourage essentially someone who has cloud credits, and if the expiration time period is arriving, just like on the initial application, I think we're very eager as a program together to continue to support cloud-based usage. So I think it's just a matter of communicating with the NIH around it.
Right, because the cloud credits are very accessible, is really a direct email to the program staff. Feel free to communicate by email with us. We do a lot of case by case. We do encourage that credits are used within 6 months. Otherwise, the account expires. But if you have like a good justification, reach out by email, so we can handle these cases case by case. And I think that, let's see if there is one more question here, and then we will move forward to the so we keep it on time. Oh, I think there are a couple of more.
Yeah. One other question is around the clinical data workflows or phenotyping. The process really is defined by the initial scope of the application and the EXO-1. So the EXO-1 investigator defines the scope of phenotypes that they have available, and it will be shared as part of the EXO-1 application. That's part of the review process. And then the way that we essentially do diligence on that process is that as data are generated prior to the PIO EXO-1 awardee receives their molecular data cohorts, we validate that they have fulfilled the commitment as defined by their application. Happy to follow up more directly, so feel free to reach out to us as well, and see if maybe the DRC can better facilitate some of this as well, Timothy.
Yeah, thank you, Adam. Nothing to add to that. All right, so let's then move forward to our next speaker, and thank you. And if you have additional questions, feel free to reach us out through emails and also in the end of this session. So I will invite now to the podium JP, who is the product owner and analyst at the Data Resource Center. JP is leading the development of the Kids First portal, and he received a master's degree in bioinformatics from Brandeis University. Thank you, JP, for being here with us today, and I will...
Thank you, Marcia. Good afternoon, everyone. As Marcia mentioned, I'm the product owner / analyst for the Kids First portal development team as part of the Sainte-Justine Research Center in Montreal under the supervision of Dr. Ferretti. So today we have the exciting news that we have a new Kids First portal. So the link here is at the bottom for the portal beta.kidsfirstdrc.org. We're happy to announce that this is a refactoring phase that we were doing in the last year or so. So we brought a lot of new features based on feedback. And with that, I want to encourage everyone. So if you want to tag along, feel free to explore it as we're doing the presentation or take the time in the following weeks to go visit it. We always encourage everyone and the community to use the feedback button on the right-hand side of your window, as this goes back directly to our team. If there's any hiccups on the portal, if you have any recommendations or comments, that goes directly to the team. We review it, and without your feedback, we actually wouldn't have implemented various upgrades with this new beta portal. One other thing that I wanted to mention is that the preferred browser currently is Google Chrome. So if everyone wants to explore that, it's probably best with Google Chrome. So let's get into it. The major phase of development was based on 4 refactoring goals. The first and main one is to improve the user experience. So what you're going to see is that you have various pages that are going to be similar and some are going to be different, and from the old legacy portal. So our main goal here was to create a more cohesive experience with the portal, to make sure that all the user interface is consistent and all the components are consistent across the platform. And we did a lot of upgrades in terms of the interface and the restructuring of it, just to make the whole experience a little bit more seamless for you all and facilitate that. I'll just move here the box in case that blocked my screen. The second major upgrade here is that we integrated the portal from a backend perspective with a Fire-powered integration. So what is Fire? It's a standard for the exchange of healthcare data information. It's also being built out in the research realm. So that's the reason why we're using it. And the reason why this is a big deal is because, as Marcia was mentioning with NCPI, so the NIH cloud-based platform interoperability effort, we're trying to leverage their Fire model so that we could use a standard process across portals. So we also work with the Include group, where this is a similar platform focused on Down syndrome, and both of these platforms use the same Fire model. And so what that does is that it creates a federated extract, transform, and load process on our end to create a type of standard way to uptake data into our portal and showcase that to everyone. So it's definitely a great win for the team. Thirdly, here we integrated a new type of expansive biospecimen representation. So I'll talk a little bit about that later. And finally, we did a lot of restructured code architecture. So this is really to optimize the portal to be more scalable, improve the data management, versioning, update our APIs to really make the platform more robust and less issue prone. And this is really going to equip us for future development, for future feature development. Now, as I was mentioning here, one of our main purposes was to improve the UI and user experience. And so what you'll see is that the styling theme across the portal will be a lot more consistent now. The tools are going to be a little bit displaced, so you're going to have to get used to that slowly but surely. You'll have a few months to get used to that. But the big bulk of the work here, and I'd say the biggest improvement to this refactor, was the pain points that we were seeing with the file repository and data exploration page being separate. So what we saw previously in the old portal is that this is a two-step process. So a user is in the Explore data page. They want to build their clinical profile of interest, so their cohort building activities start here. And then once they figure that out and they're satisfied with their virtual cohort, then they're redirected to the file repository here by clicking on this redirect. And so what that does is that it leads you to the file repository as a separate page. And then you could also continue your querying activities on the left-hand side. But the extensive searchability is not the exact same as the Explore data. So the Explore data is really the page where you're meant to build that virtual cohort and identify your files of interest. And there are also various features that weren't available in this file repository, such as saving your file set. You couldn't combine queries, etc. So we really wanted to update that. And so what we did is that we integrated the file repository into the data exploration page. So you'll see here, this is really going to enhance the query building activities for something that's a lot more natural and streamlined instead of having this two-step process. So what you see here, you have the facets on the left-hand side. You have the facets for the different categories. And what's interesting and the powerful part of this is that you'll see here you have your participants. So for the clinical demographic background, your biospecimen metadata, and your data files. So what you saw in the separate file repository page is now seen in the data files tab here. So what that allows is that it creates a much more synergistic experience as you're applying filters. Is going to update each and it's going to update each of the three entities, the participants, the biospecimen, data files, simultaneously. So what this really allows is that you can now see at a row level the equivalence between each entity, and that's something that was a little redundant in the old version, where you had to go back and forth between the Explore data and the file repository if you need to do some adjustment to your cohort building activities. So this is a very big win. And you'll also notice that we now have a biospecimen tab here. So I want to dive a little bit into that. So this is part of the visualization that's improved for the biospecimens. So you're going to have all your metadata for your biospecimens here within a tab. And previously, that was only done through biospecimen data modalities, through facets. So you could search through the facets, which you could also do now. And you could only download that information in a tabular view through a CSV report. But now we have, this really facilitates the visualization of those associations between the clinical part, the biospecimens, and the data files. So for example, if you're looking for CRAM files that are associated to tumor samples, well, then you could do the one-for-one between the biospecimens and the data files here live during your cohort building activities. We also added the identification of sample availability. Currently, they're mostly set out and available, but down the line, this may be something that will be considered during data ingest. And finally, we improved the navigation across the sample hierarchies. So what I mean by that is, as I was mentioning earlier, we updated the biospecimen data model. So what I'm going to, without going into too much details, what we have here is a sample collection event ID. So this is usually associated to a primary collection of blood, urine, saliva, tissue, etc. And this is really going to englobe and encapsulate all the samples within this collection event ID. So what you're going to have here, you'll start off with, for example, a blood sample called sample number 1. And this blood sample could go through 2 routes. There's also other routes, but I'll keep it in a simplified view for now. So this blood sample could be aliquoted in 3 different aliquots, and these are going to be labeled as containers, and they'll have their own container IDs. And these are going to be biologically equivalent, assuming that there's homogeneity in the blood sample. The blood sample could also undergo a processing or treatment, for example, a type of centrifugation to isolate white blood cells. And through a type of derivation, the biological nature of the sample changed. And so what happens in this data model is that creates a second sample, as you can see here with the orange. So the white blood cells is now sample number 2. And the concept that we really want to showcase is now you have the concept of a hierarchy. So you have a parent and child relationship between the blood sample 1 and the white blood cell sample 2. And so the same thing could happen in sequential events of times. So the sample 2 could undergo aliquoting as well. They'll create aliquot 1, 2, 3 that's associated to sample 2. These are all going to be biologically equivalent. And similarly, if you want to isolate, extract and purify the DNA from this white blood cell sample, then you're going to get your DNA sample number 3 because the nature of it changed again. And this is, again, in the context of Kids First, what it's going to be sequenced and sent to us and analyzed. So again, showing here the hierarchy and the sequential hierarchy of the sample. So again, the parent sample of this DNA is going to be white blood cells and so on, all the way, and that's captured across the whole lineage of this sample derivation and aliquot process. So this is going to be really cool once we're going to have the integration is complete for the long read pilot and the proteomics intake. So the proteomics is going to complement the whole genome sequencing and RNA-seq data. And with the new and more flexible model, then it's going to provide a little bit more information on that associated biospecimen / clinical information. So that's going to be great to see. Now, the last step for the data exploration page, we have now a new summary view that's a little bit more intuitive and interactive. It's going to carry a more customizable layout where you can increase, decrease the size, move around the cards, and all of this is going to be saved to your profile. So once you're going to come back in a subsequent login, it's going to still be at the same position and the same configurations that you've done previously. Another feature is that you can now save the charts as images in PNG or SVG format, and you could also do the same for the TSV data. So this is just a nice add-on for flexibility and searchability of data through, instead of using the facets on the left-hand side. Currently, it's limited to some of the facets, but we're planning on expanding that to have most of, if not all of the facets in the portal for the more visual users. Another major upgrade here was with the germline variant exploration tool. We added various new annotations such as the PICT, or the most deleterious mutation consequence, is the one that's showcased. What I mean by that is that now, I'm not sure if you all could see my mouse, but in the table below, you could see that there's a most deleterious consequence column. It used to just be the gene consequence column, where it listed all the consequences associated to the variant. But now we really want to make the most deleterious consequence pop. It doesn't mean that the previous and other consequences associated to the variant aren't available. You can, as you're applying your facets, et cetera, for certain consequences, it's still going to show the relevant variant. And you could also see that in the variant entity page. So it's just a great way to highlight the most impactful consequence. Other annotations that we added is the NOMAD PLI and left score. So these are prediction scores to see what the likelihood of a what the likelihood is of a variant to be protein truncating and thus causing a loss of function. Similarly, we also added the Splice AI score. So this is, again, is a prediction score to see the likelihood of a variant affecting a splicing event. Finally, we upgraded the NOMAD 3.11 to NOMAD 3.12 just to be with the latest data that's available in these public databases. Finally, another big piece that's not necessarily seen on the UI portion, but on the backend side, we really improved the scalability and the variant data management / versioning. So this is because the variant data that you see in the portal is stored in VCF / and these VCFs are stored in Parquet files previously. But now we've made a modification to ingest those into Delta Lake tables, which are much better for data management and versioning. So this is really going to empower the ingest of other studies down the line. And we've also restructured the variant gene and occurrence indices within Elasticsearch, which is essentially our search and queryability database for large-scale data. So this is really going to empower scalability at the end of the day. Now, for upcoming features, we're going to be, and this is more so from the short term of things, we're going to be implementing a study entity page. So this is really just to provide users with more detailed information on each study as it's done for the participants and the files. We're also going to be implementing, as part of the variant exploration page, a type of phenotype aggregation. So what this is going to do is going to generate an overview of the participants' phenotypes based on a set of selected variants, of course, in aggregations. So this is going to facilitate possibly disease association studies, or generally just having a better understanding of biological mechanism based on that phenotype to variant association. Next, we're going to be working on the quick search. So this is again just a type of quick improve the search and create a type of text-free input where you could search on various facets or facet values during your exploration activities. And finally, we're also going to be working with CAVATICA for a variant workbench where you could query, mangle, analyze, and visualize kids' first genomic variants from participating cohorts within CAVATICA's cloud compute environment. So that's going to be very exciting. And finally, the last piece here for upcoming features is once we've adjusted, you'll see that we adjusted the dashboard to be a little bit, again, more customizable. It's going to be more of your workspace environment where you could save your saved queries, your sets, etc. But the piece that I want to highlight here is that we want to integrate a type of analytics section where you could have a history of your analyses, so you don't have to re-enter your parameters every single time for each analysis. And we're also going to be adding some of the new and more relevant analytic tools within the web-based application to facilitate users to stay within this, again, one workspace. So this could be like an enrichment analysis, a gene expression analysis, or comparison across cohorts that you can build with some of your saved sets. And that generally covers the additions that we've done to this new refactor. So again, I encourage everyone to go take a visit to this new portal. We're going to be phasing out the old portal within the next following months. So again, familiarize yourself with it, play around with it, and don't hesitate to let us know what your feedback is or your comments as well. So David's going to be giving a little bit of a live demo of that. So I didn't cover all the upgrades. So again, go take a visit, and David's probably going to highlight some of those now, and open to any questions as well.
Thank you, JP. And we may leave the questions for after David goes, and then you two could answer all the questions about the portal. And I just want to quickly introduce Dr. David Higgins. He's a program manager of informatics at the Children's Hospital of Philadelphia. He brings experience not only in genomic sequencing and genetic variant identification, but also he is bringing his experience in training and communication. So David, it's all yours.
Lost the mute button there. Thank you so much, Marcia. Yes, so thanks for that great kind of list of all the different features and highlights that JP shared. And so I get the really fun job because I got to do the live demo. And so I'm going to pull up our new updated beta portal for the Kids First DRC. And I'm going to show some of the new features, some of which JP mentioned, and I'll highlight a couple things that Marcia talked about at the beginning of her discussion as well. So I'm going to exit out of my slides. And I'm going to switch over, so I'm going to navigate to this URL, and I've already got it pulled up. So this is portal-beta.kidsfirstdrc.org. And so I believe, from speaking with JP, I believe we're going to have a kind of like a banner at the top of the current portal that just lets folks know about the new one. And then eventually in the coming months, we are going to switch those over. So then eventually Portal Beta will just become Portal. And then this will be the new Kids First portal in the future. So when you come to the dashboard page, you'll see some similar features, but underlying this, there are actually some improvements as well. And so one I want to highlight is the CAVATICA connectivity. So in the past, when we were connecting to CAVATICA, you had to navigate to CAVATICA, find this ID password token, and copy it and paste it back to the portal. But we've actually improved that now. And so you'll just be able to be redirected to log in with CAVATICA, log in with your ERA Commons ID, and authorize Gen 3. And then it should send me back to the portal, and everything will be reconnected. And so there are my different demo projects that I have in CAVATICA. So it makes it easier to move files from the portal to CAVATICA, which is our main use case that we're always trying to share. I know that Sean is a user who's using CAVATICA a lot, and he'll share some of his tools after this talk. All right, so that's one of the features that we've built. So other things up here is to look at the top. And so as JP was kind of mentioning, is that we used to have Explore Data and File Repository were two separate tools. And so the big change is that we've compressed those down into this single data exploration table. So before we go there, I want to look at the studies page. And so the studies, what we've done is we've really kind of simplified the studies view. And so for all of our different Kids First data sets, of which we have 30 right now in the beta portal, it lays out a different code, these little short codes. So these will pop up throughout the website, but they all map to a full study name. And then we have additional information as far as where to receive access and what types of data are available for the particular study. And so Marcia mentioned that, for example, one of our new studies that we released this year is this genomic analysis of laterality birth defects. And so if you're interested in learning more about it or applying, it links out to dbGaP right here, this PHS number. And so if we click that, then it will pull up the dbGaP page, which gives you more information about the study, but also it gives links about the consent group of the patients, the number of participants in the study, but then also gives you the opportunities to be able to apply for access. And so this is how you receive access to the controlled access Kids First data. And then once you are approved, then you'll be able to use those files on the Kids First portal and copy them over to your CAVATICA project. So everything links back to this space on our studies page. But before you do that, maybe you want to see, well, what types of data is in there? Let's check out a little bit further. And so this tells us that for this laterality birth defect study, there are 739 participants. So let's learn more about them. So if you click that 739, so now we've moved over. Now we're in the third tab up here. We're in data exploration. And so it's already pulled up a query. It's added that code. And so now I'm just looking at those participants in the birth defects study. And so this highlights, it presents it here at first in this kind of table view with these 739 participants. And it gives information about some demographic information about the patient, but also shown here, which I really like about the new user interface, is that it really highlights the phenotypes of the participants. And so this is work that the data team has been working on a lot this year, is how do we get more data out of the submitted clinical data sheets, and how do we get more of it onto the portal to allow users to be able to use this information to query and find the genomes that are interesting to them. And so this particular study has really deep phenotyping. And so some of these individuals are not probands, but I can filter that down. So I can go, my filters now are in the left-hand column, and I can select just the probands in the study. And so now I'm just looking at 526 participants. And now you notice that the proband column is always true. And so we use the human phenotype ontology, HPO, to harmonize clinical data across our different studies. And then it gives you information, for each of these terms with the kind of the plain text code as well as the HPO code. And so as you're scrolling through here, you may see something, that you're not exactly sure what this means. And so you may see a code that, well, I don't know what that is. You can actually click each of these codes as a link. And so you can click it. And it tells us, oh, this is also called clubfoot. It has 4 different components. It's a development error of the foot and bones of the lower leg. So it gives you information about what those codes are if you need a little bit of help, kind of understanding what all these different ontologies mean. So what I really like about highlighting all these different phenotypic data is that it really allows for and demonstrates this cross-study comparisons and analysis that the Kids First DRC is really trying to promote and drive. And so, for example, you can see that this participant here has abnormalities of the kidney as well as one of their ontology codes. So I could, maybe I'm interested in kidney defects and I want to learn more. And so I will start a new query, and we can search for that specific ontology code. So I would go in the left-hand column and click on clinical data. And then choose a observed phenotype HPO code. And so what it's doing is it's loading up this big kind of table or list of all the different HPO ontology codes that we have in our database. And so I can type in kidney. And so HPO is a hierarchy, and so there are very broad general terms like abnormality of the kidney, but then there is also very, very specific terms as well. And so you can select at multiple layers to be able to capture very specific or very broad number of participants. So for our demo today, I'm going to start very broad, and I'm going to choose abnormality of the kidney, and I'm going to add that to my query. And so now you can see that's popped up here in my box. And so now what we're looking at is there are 449 participants in the database that have either abnormality of the kidney or any more specific term below that. And so that's what you can see from scrolling through this list here of the different ontologies, is that, you know, some individuals have very specific kidney conditions, whereas others might have very kind of general kidney conditions as well. So if I want to learn more, you know, I can scroll through this great table. We can select all of these files, and I could download the clinical data for all of them. But there is, as JP was alluding to, there's also a summary view, which provides kind of an overview, you know, pictorial, graphical representation of all of this stuff. And so that's always available right here to the left, this summary tab. And so if I click on Summary tab, it provides lots of different charts here with different pieces of information. And so first up is the studies kind of pie chart or table. And so it tells us about the different studies that these conditions, these participants were enrolled in. So KFKUT is kidney and urinary tract. And so, you know, that kind of is what we would expect, that a lot of participants in that study also have abnormalities of the kidney. But by scrolling through the others, you know, KFCHD is congenital heart defects. And so there's 51 participants with the heart defects that also have a kidney defect. Likewise, EATF, that's the esophageal atresia study. And so you can see kind of the connection across all these different studies that, you know, if you're coming in interested in kidneys, check out the heart defect study, check out the esophageal atresia study as well. And so, you know, really kind of making all these different data sets available to try to empower and expand your analysis to hopefully improve your statistical findings of the things that you are researching. So below, and I guess we have additional graphical displays of the different phenotypes. We have different graphical displays of the demographics of the participants, as well as different data types, so the files that are available for these participants. So you might be looking in this, and it's all these bars are all a little bit lined up and out of skew here. So the really cool thing about the updated portal is you can kind of make this summary page your own. And so each of these boxes can be dragged around. You can shrink them, you can grow them. We can put this one up here and put this one over here. And so they all kind of move around in their own way. And so I really like this because I find that I'm usually hopping between different monitors, different setups, and sometimes I have a tight resolution, and sometimes I have a huge resolution, and then everything kind of shifts on me. And so now you can make it exactly what you want, you know, however you want to work on the portal. So if you're really interested in data types, you can make that graph huge. If you're really, you know, not as interested, you can shrink it or even exit out. And so it really makes this kind of interactive way to really explore the data and see what's in here. So we're just getting started with these different facets of how do we kind of improve these and add new facets as well. But this is one of the exciting new features. So we're really excited, this summary view table. If I have a couple more minutes, which I do, so I'll also share a new query. And so I've been using our ontology codes so far, our HPO ontology, to do my queries. So I'm going to do a new query, and I'm going to highlight a new feature that we built in related to our other ontology, the Mondo ontology. So Mondo is our diagnosis ontology. And so if I go to participants again, and I go to clinical. And so before, the Mondo exploration had a very kind of small search box. It was not very user-friendly. And so now with the improvements that the portal team has built, now Mondo gets its own pop-up just like HPO does. And so Mondo, also like HPO, is that it also has a hierarchical structure. And so we can search for a disease. For example, we could search for leukemia. And of course, there's lots of different subtypes of leukemia. But if I want to select, again, for the sake of my example here, I'll just select a very high-level term of leukemia. And then it allows us to be able to see the diagnoses of each of these participants as well. So this individual, for example, this top participant here, they have ALL leukemia as well as trisomy 21. And so we can go back to that summary page. So now you can see my graphs are all in the same spot that I left them, but now they all have different information. And so the studies here for my leukemia diagnosis is that most of these patients are in the T-cell leukemia study, as well as the Down syndrome and leukemia study. And so again, it makes sense that we've updated all of these graphs as we've changed. And if we switch over back to my kidney, then you can see all the graphs change again. So the summary table will refresh depending on what query you have active in order to be able to explore. And so then the last feature I'll show on this page is that I'll highlight. So we've talked a lot about clinical data and using clinical data to search for files, but ultimately, we want to get to the files. And so that's how do we explore those? And so we can switch over, back and forth laterally between the participants, the biospecimens, and the data files. And so if I go back to the kidney query, you can see that we have many different files available for these participants. And so I can explore and add, for example, based on data type. If you are interested in the alignments and you want to start from the very beginning, you can search from alignments. If you are interested in, for example, the GVCF, the Kids First DRC produces harmonized GVCFs. It can be used to be combined together as part of a joint call analysis. So if you're just interested in those GVCFs, you can add that checkbox and then apply. And then you can see now we're looking at all these VCFs for these different patients across these different studies, all of whom are labeled with a diagnosis of abnormality of the kidney. So lots of different ways you can also get here from the summary tab. And so you can see my summary tables have updated, is that now we're just looking at those GVCFs because I've added this filter here. But you can also click and explore these tables as well. So if I back out and let everything refresh that query one more time, then I should be able to click that GVCF button and we'll get back into business here. All right, so I'm going to let that load a little bit longer, but in the interest of time, I am going to move on. And so the last piece I'll show is our variance table. So variance is a feature, there it goes, it finally loaded. Variance is our feature that we launched on the portal a couple years ago at this point. And so it allows users to be able to search using the genomic variants of interest. And so, similar to how data exploration allows users to be able to search across phenotypes, search across diagnoses, across the different data files. The point of the variance is that it allows for searching based on the genetic features. And so the portal may need a moment to get back up because it is not loading the data correctly right now. But at this point in these variant search on our portal beta, we have loaded, I believe, 3 or 4 different studies that highlight some of those features that JP mentioned. It works very similarly. It does a nice kind of condensation that we are now just looking at the most deleterious consequence of the portal or of the particular variant. And so that kind of makes a neater presentation as well. So with that, I'm going to try to refresh my screen here and see if I can get some variants to pop up. But I think JP and I are happy to take any questions from folks in the chat.
It may just be that the beta instance is undergoing huge hits right now.
Yeah, you know, I was thinking about that perhaps people are so excited to get into Portal Beta that now there might be so many people in here that I can't show Portal Beta. So that is a possibility of something that we were afraid of. So let me log out and log back in, and I'll see what happens. But other than that, I'm happy to answer any questions while we're waiting.
Right, and JP, if you want to come back to the podium, I think there are a couple of questions here. Let's see, Claudia Carvalho, if we want to confirm bioinformatic findings with orthogonal methodology, is it possible to request biospecimen, or should we discuss that with the submitting researcher directly?
Yeah, that's a great question, and it's actually a use case that we are piloting right now. So as you suggest, right, we can provide a really facile framework for a biorepository to potentially indicate whether a sample is available or not available for request and facilitate that process with a biorepository. So we're actually in the process of piloting that framework where a user can create a cohort not only of data, but of samples, and then interact with the repository or investigator that owns, quote unquote, that sample for downstream research, bringing to that context, a manifest of the samples of interest based on their availability. So that'll be a feature you'll see very shortly. We're piloting it with one repository and then extend that.
Okay, we have another question. In the exploratory mode prior to download, can you download a manifest that links the file? Sorry, I lost links the file name to the patient ID.
Yes, you should be able to do that under the data files page. There is an opportunity if you select, you can select all, and then there's an opportunity to download a manifest. This is on the data files page, and then that would link it back to the appropriate ID. Great question. So I did also want to highlight, I was able, the best tech support there is, turn it off and turn it back on again. And I can see the variants now. So just to highlight really quickly, variant search and the kind of the updates to the UI here. You can search based on a couple different facets. You can search if you're just looking for participants in a particular study, you can. So at this point, we only have 3 studies loaded in the Portal Beta, but we're looking to add more before the full release later this year. And then you can search if you have a very specific variant, like a DB-SNP number, you can, or even a specific coordinate in the genome, you can search as well. I usually search by gene is the type of the demo that I usually show. And so most folks have a favorite gene that they are interested in researching. And again, I'm having trouble here finding some of my favorite genes. So I will say the Portal Beta is not hooked up to the same computer motor that the big current production portal is set up. So it does not have enough juice under the hood to use all the folks who want to get on Portal Beta right now. So we are going to continue to work on that in the future. But it does highlight a lot of information as far as Nomad, different studies and things like that.
And David and JP, when we should expect to have the portal on production for all?
So it's currently open to the public, but we're going to be ingesting more data over the next following weeks. So it's going to be a little bit more complete, as David was mentioning, matching what we currently have in the legacy portal. The variance is very big data, so it takes a little bit of time. That's why I'm saying it's in the following weeks. But this URL is available to the public currently if anyone wants to familiarize themselves with the user interface, etc.
Yeah, so to be clear, both the beta portal and the existing portal are currently available. And I'll touch on this at the end, but one of our main goals by going ahead and releasing it, even as we are still loading data into this context, is to make sure we get feedback from the community who's already familiar with and is using the portals so that we can modify or edit or further inform as we're beginning to reach that milestone of, final portal release. So please take an opportunity to explore and further inform and give us feedback, making sure that we captured all the features you loved and features that you want to have, as we move forward.
Thank you, Adam. And as I want to take Adam's actually last sentence here and say how important is your feedback, because the Kids First program is where it is today. And we have that sizable data now available because of your collaboration, participation, collaborating with your feedback and your work with us. So we really appreciate that. So we will then, if there are no other questions that I may have missed, and sake of time, our next speaker, Sean, I would like to call Sean Griffith. Hi, Sean.
Hello.
Welcome. Sean is a senior bioinformatics and software engineering at Johns Hopkins Hospital, and we will talk about a new tool, PhenoDB, that is connected with CAVATICA. So, Sean.
Hi, everyone. So I'm presenting these slides on behalf of Laura Vale, who's actually a software engineer that works on my team at Johns Hopkins Genomics and in the Sabrera lab at Johns Hopkins University. So if I stumble a little bit over the slides, you'll have to forgive me. Laura prepared these for me. Before we get started, I just want to talk a little bit about what PhenoDB is. So PhenoDB is a web-based clinical research tool that was originally developed as part of the Hopkins and Baylor Center for Mendelian Genomics, which was an NIH program. This tool has been under development since 2014 and sort of actively used, for 8 or 9 years here. This tool is available to the public at phenodb.org. It's a web-based tool, and there are a variety of installations that people have sort of globally, and then there's the main installations that we have at Hopkins. People are welcome to host their own instances of the PhenoDB platform as a web-based tool. As sort of the managers of the PhenoDB project from a technical perspective, we had the issue that there were Kids First collaborators who had their data on CAVATICA, who wanted to perform the analyses that we had in PhenoDB, but without pulling down their data from CAVATICA and then uploading it into a different system. So we started working with the CAVATICA team to take some of the PhenoDB analyses and integrate them into CAVATICA as a tool that people who are already CAVATICA users could take advantage of. So we're essentially taking advantage of CAVATICA's data security, storage, and computing power to perform some of these analyses that our collaborators that are part of the Kids First program wanted to perform. To use PhenoDB on CAVATICA, all you need is a VCF file from an individual with suspected Mendelian disease, and optionally VCF files from their relatives. Obviously, having relatives helps narrow down possible causes of the disorder. So what PhenoDB does is it runs ANNOVAR to annotate variants that are present in the VCFs, and then analyzes the individual's effectiveness, relatedness, and other characteristics to help identify candidate variants that might cause the disease in question. So before I start here, I just wanted to say too, and I'll get into this a little bit more later as needed, but PhenoDB, the app, is available on CAVATICA, but we're currently dealing with an issue that the CAVATICA team is aware of. So the app is essentially undergoing some maintenance right now. We expect it to be up and running normally within the next couple of weeks, but I just wanted to let everyone know that if you go there to use it today, you might not be able to do so successfully, which is why I have these screen grabs rather than a live demonstration of the tool. But essentially what we have here is the 2 steps as separate CAVATICA apps that are run in sequence. First, ANNOVAR annotation for PhenoDB, and then PhenoDB analysis using those annotations and VCF files. So yeah, you first run the ANNOVAR annotation app with all VCFs you want to analyze, and then analyze the annotated files with PhenoDB analysis. And let me just show you what that will look like. So this is the user interface for this ANNOVAR annotation app. It lets you select samples from the CAVATICA space, and then also what annotations you want to perform using ANNOVAR, and then a human assembly. So this supports 38 and 19. So resulting from that, we have these essentially text files that contain all of those ANNOVAR annotations that will also be stored on CAVATICA. And then you use those to run a PhenoDB analysis. When you're running that analysis, you can select several analysis types, minor allele frequency, what ref gene location you're interested in, and then what your sample statuses are. So affected, sample sex, things like that. These are just some of the inheritance patterns that we're able to use during this PhenoDB analysis. And these are the required characteristics for each individual. So what relation to the proband we have, the sex of the individual, whether they're affected or unaffected by the Mendelian disease, and an annotated VCF file from that first step. And as I pointed out before, you can also include minor allele frequency that you're interested in and a ref gene location. From that analysis, we get a tab delimited file that contains information that the researcher can look at and use to determine possible candidate genes or candidate variants that may have caused the disease. And working with our friends at CAVATICA, we were able to just present that information directly in CAVATICA. Users don't have to download it and look at it. It's sort of directly interactable with here in the CAVATICA GUI. And there's also a detailed log mirroring what we get out of PhenoDB itself that goes through all the different analyses that happened during the PhenoDB analysis, all the different steps that occurred. And this is useful for troubleshooting, or if you want to recreate any of these steps on your own. Obviously that's not quite as interactive as if the app were currently functional, but hopefully we'll have it functional within the next couple of weeks. And like I said, we're actively working with the CAVATICA team. Hopefully some of you are familiar with PhenoDB and have used it before, but our goal is to essentially recreate PhenoDB analysis as part of the CAVATICA workspace to enable combined users. Are there any questions about that?
Thank you, Sean. Any questions for Sean? No questions here, but as the questions come, we can also take some questions in the end of the program. And I would like to thank you, Sean, for the demo, and also JP and David and Sean for all the demos. As I said in the beginning, the goal of the Kids First public webinars is really to help you and point you to explore the resources we're building as part of the Kids First program, but also tools like the PhenoDB that was a outside development and now is connected with Kids First. So that is a great example of collaboration, not only within the program, but with outside investigators and researchers. So thank you again. And I think we're gonna move for our last speaker.
So just
Adam.
one, if you don't mind,
Go
just one
ahead.
quick comment too, for people that are considering moving or adding their app or workflow to CAVATICA, I highly recommend it. We did find the CAVATICA integration process and the CAVATICA team to be much more receptive than average for these kinds of tools. So just, you know, throwing that out there for people who might be considering doing what we did and moving their tools into the CAVATICA environment.
Thank you, Sean. And so we will move now for our last talk of the day. Adam Resnick, Director of the Data-Driven Discovery at the Children's Hospital of Philadelphia. His expertise in pediatric brain tumors and translational research. He will talk about his vision for the Kids First Data Resource Center. So Adam.
Thanks so much. And again, Sean, you know, I just want to give kudos to you and your team for sort of really taking that on and sort of building on, you know, what is a fantastic environment for variant annotation and case solving and bringing that into, you know, the CAVATICA framework in a way that potentially provides, you know, a seamless framework for moving from, you know, portals to exploration to then annotations and discovery. You know, I think we're really excited sort of by your team's sense of adventure in bringing that into the platform. You know, so what I'll do, and I know we're a little bit behind, is really just touch on a couple of points to really frame where we are, you know, with this new portal launch and what does beta mean after all. So one, beta means that, as Allison put it in the chat, that you shouldn't give the link until after you presented so that we don't get hundreds of people using the beta instance all at once during the demo. But to me, this is a sign that there's a ton of interest in all these features. So please continue to explore and provide feedback, you know, post webinar. And really, you know, our goal is to sort of break and test, you know, over the coming weeks to make sure that as we transition from the existing portal into this new portal, we really are both advancing the current users' capacity, then engage further. So give feedback. Don't be shy about giving feedback for this. But the other part that I wanted to highlight, besides this sort of front-end feature sets that you've seen to enhance portal usability, is really the direction we're moving in to support you know, new ways of thinking about the infrastructure and workflows associated with the Kids First program and portal. So JP mentioned, you know, the biospecimen data model, and I wanted to contextualize this and maybe touch on and relate this to the questions that came up, which I think is fantastic because it highlights, at least to me, that we're on the right track of where we're heading. You know, as was mentioned, you know, our initial Kids First workflow was really, you know, an EXA1 awardee, you know, sent their samples to a sequencing center, whole genome sequence and RNA-seq data generation took place, and the data resource center ingested, you know, that one time point of whole genome sequence and or RNA-seq data as a single instance. So what's changing and why do we actually have to address, you know, the biospecimen model? And so the really key part that we're seeing, which is I think natural as part of the scientific workflow, is that, and I think this is really, you know, a very exciting component of the Kids First program as a whole, is that investigators themselves are undergoing multiple rounds of data generation and integration. And this is in alignment with the multimodal expansion of Kids First. So just as a framing, you know, a number of investigators, you know, had a whole genome sequence and RNA-seq performed, short-read whole genome sequence. Those same investigators then were awarded a resource for generating long-read sequencing data. So now suddenly you have the same sample undergoing whole genome sequencing and then the long-read data generation separately. And then more recently, you know, similarly, the investigators have had whole genome and RNA-seq generated under Kids First now have proteomic or metabolomics or post-early modification data generated. This is a natural course of the scientific process that we really want to tie into the Kids First platform. And so in order to accommodate this asynchronous data generation across samples, the same samples, different aliquots, and tie the data to the right sample, we had to really invest in the new model and its deployment. But I don't want to miss, the opportunity that was highlighted by the question around the idea that once you represent samples within a portal, their availability status and quantities, concentration, and length into data, you also open up the opportunity for collaborations, not just by that single investigator, but by external partners who might want to essentially suggest or engage the use of that data and samples for additional studies. And I think that's a really terrific sort of next step of where we want to move to, is to empower collaboration across the analog space to create shared digital artifacts through this iterative scientific framing of use. So I think we're really excited about that framework to move forward. JP mentioned FHIR as a key change, with the portal really undergoing a sea change of the underlying infrastructure and resources being encoded as a unified model represented in FHIR. And I wanted to contextualize this a bit further because I think it's a very timely development. And to really expand the notion that it's not just about a common model, but also what does it get us by really operating in this FHIR framework, and set the stage for what I think is a really exciting next phase of activities, which really provide the opportunity to create a standardized, automated, more comprehensive and longitudinal approach to clinical data and its incorporation into the portal. So what is the current model? So very similar to the biosamples, the current model is really based on investigators who enroll, consent, and abstract data, from the electronic health record, and then submit that data as a single point in time in association with the samples that they submit. So right now, there are still limited ways for us to continue to update that data if, for example, that participant continues to be followed by the study team. And the study team itself is limited by the sort of overhead of data curation and abstraction that they have to undertake, because by and large, most case report forms are being fulfilled manually by the coordinators or study team in ways that is challenging to do longitudinally and across time. But I think that effort of curation and submission really highlighted a couple of key points of opportunity for us to intersect with in the context of emerging FHIR technologies. And specifically, this relates to the data workflows for FHIR. And so by shifting the Kids First portal in its entirety into a FHIR underlying infrastructure, we're really aligning with what is a federally framed setting for data exchange that's linked to the 21st Century Cures Act. And this is really a process driven by the Office of National Controller, where this past year, beginning in 2023, there's really been a change in the way that the electronic health records and systems that capture clinical data operate. And so here I just took a snippet from Health IT Buzz and their reflection on the sea change, right? So they say, and I'll read it briefly, the moment we've been waiting for since 2016 are fast approaching. The 21st Century Cures Act infrastructure is coming into place in 2022. This was last year. The Cures Act laid out a vision for a health ecosystem of standards-based APIs and a national or nationwide health information network that supports the exchange of that data. And importantly, that framework is underpinned by the certification provided by the federal government to clinical systems that support the use of FHIR APIs that changed this past year. So what does that mean? It suddenly means that the source of the data is adopting a new standard for the exchange of that data. So most of us who collect specimens and associated phenotypic data begin at the healthcare framework. And having FHIR as an anchor to start with provides a new framework for us to operationalize, iterate, and develop tools, not just for the portal itself, but for the ongoing operations and exploration of data. So this is an ongoing pilot that we are performing as part of Kids First, where we're beginning to pilot the use of FHIR messages from electronic health records in partnership with AWS and the Amazon Health Lake team to support that ingestion of data more directly from electronic health record systems, processing the identification and ingestion and integration initially into that REDCap form. So the question that we can ask is, can we reach 80% of a CRF being filled out automatically? And can that be a tool that can be deployed broadly across studies for those who have permissions to support that framework in ways that can provide longitudinal, more real-time data integration? The other exciting framework that this brings into light is capitalizing on that structured framework of FHIR and the toolkits that come on board by bringing in this API-based framework into existence, is that we can start layering in and piloting efforts around the use of large language models and ways to standardize and model the data, even from an unstructured framework as represented in FHIR. And this is an ongoing pilot that we'll be engaged in with this year. So that combination of essentially, a model that spans biosamples, clinical data, and research-derived sample data sets as a queryable framework that reaches further into the source setting and develop tools and workflows to integrate them into a queryable space, as well as tools that operate on those data and metadata in ways that empower users to interact with them through the portal, but also outside of the portal through other tools, is a key component. And that's a key part of that last piece that JP mentioned, which is this restructured code architecture and the APIs. So heavily investing, for us as a CRC in the API setting of the underlying infrastructure so that other tools can be developed around these workflows. And importantly, that we can begin piloting a more virtuous cycle of discovery. Up until now, and most of the publications you'll see about Kids First focus on these cohort-based discoveries, which I think are terrifically empowered by the cross-disease setting of Kids First. But that cohort-based discovery now is beginning to provide an opportunity for that translational loop to completion. You're getting a hint of that with folks like the Hopkins group deploying PhenoDB into CAVATICA, because it provides PhenoDB, right, as a mission of its structure is really supporting the solving of individual cases through their annotation. Solving individual cases through annotation is linked to these large cohorts, and their unified processing and harmonized phenotyping is the key next use case. And to get there, we really have to develop and empower these API structures so that these types of activities can happen within CAVATICA or by other systems that can interact with the API endpoints of the Kids First setting. But for us, this is going to be a key area of further development in this coming year. So these are just three key drivers, for these beta updates that we want you to keep in mind as you provide feedback. One, just maybe a fourth would be one, just a look and feel and ease of use, of the portal as you interact with it. The second is, we're incorporating this biosample-based model. As was mentioned in the chat, some studies will not have very deep biosample metadata, but we're taking the opportunity with this refresh to bring in additional deep biosample data, especially for cohorts that have multiple layers of data generated for them. And then, as I mentioned, doing a pilot around linkage of biorepositories and their queryability and requestability of samples in that context, and linking them to both the data and participants. And then the last piece is, this intersection of an API-enabled framework that tools and other resources can connect to in ways that can both support cohort-based discoveries of the phenotype to genotype correlations, but even more importantly, begin to pilot efforts where these types of resources can drive so that feedback loop into individual solving of cases in a much more translational set way. And there, really, we are investing heavily in FHIR-enabled technologies, working with a number of technology partners like AWS to support that very deep longitudinal clinical and outcome-based framework that can be empowered by a much more real-time framework for data submission, curation, and integration into the underlying database and ultimately the portal. So we hope that this provides a framework for you to then provide feedback, ask questions, and further inform the beta version of the portal as we move into that next phase of resources provided by the DRC and the tools that we support. Thanks again for joining today.
Thank you, Adam. And I don't think there were new questions. I think the questions being, and so let's give a little bit of time. So how long you envision to like the portal be fully integrated? To have it?
For us, we hope to complete data load and sort of scale compute and environment over the next month. So that essentially you'll have the same representation of data across the existing portal and the new portal. And that'll be just the first, essentially, base camp, for that. During that period of time, we also hope to get feedback, from the community. We'll also be, as a team, at the ASHG meeting there in person. So please consider coming to the ancillary meeting and further inform more directly with us and ask questions at that meeting coming up. I think there's a combination of both, bringing the portal, the beta version, to completeness, and then collecting user feedback, questions, and guidance is going to be a key next step for us.
Right, so any questions from the audience? I think we had lots of questions that we could answer during the talk, but this was great. I think there is a couple of, so thank you so much, Adam. Kids First is really setting the stage for data sharing, empowering the scientific community to work together in rare diseases and pediatric cancers. Congratulations, team. We also got a comment from, so there's comments and answers to other questions. Yeah.
I think these kind of questions are really great to see. I think they're, really detailed and really represent, a deep engagement by those of you who are on the call. So please keep it coming and make sure that you can continue to inform. Essentially, you informing it will change what we do as we move forward. And like I said, we really want to set the stage for the next iteration around both cohort-based discovery, solving individual cases, and optimizing on the longitudinality and data workflows that can really be empowered for discovery and linked to biosamples. These are areas of focus that we really want feedback on as we further launch the portal.
Wonderful. Well, thank you everybody for your engagement. I was really engaging session with lots of questions, and thank you for the Kids First staff and the Kids First Data Resource Center staff for being here and helping us to answer the questions. Thank you, Alison Heff, Andrea Harris, JP and David again, and thank you, Adam, for your vision. We will be back here on the spring, but between now and then, we hope to see you at the annual meeting of the American Society for Human Genetics that will be happening in Washington, D.C., and we will have an event. So thank you again for the great session today.
Yeah, thanks so much. And please provide also feedback. I think you'll get either via email, I see it in the chat, a survey about today. Look forward for additional input. Thanks so much.
And I see the survey now, so I will stay here a little bit so people that are on the call still can see the survey. We ask you to give us feedback through the survey, and we're also going to follow up by email. So thank you, Ada, again. And with that, we will close the session in a minute or so. Thank you.